there. And it sits in these reservoir populations, and the main reservoir population is this animal here. It's called a great gerbil. The Russians are very proud of their great gerbil, and it's a decent sized animal. It's a decent sized animal. It's not as small as a mouse or a rat. Maybe it's about the size of a possum, maybe a little bit smaller. But these animals are a fantastic reservoir for the plague bacteria. Some of them are very resistant to plague, and they don't have any symptoms, but they carry the plague. Whereas others are really affected by the plague and die quickly. So there's this really heterogeneous response in the animals when they get infected with plague. That helps the bacteria spread, and it helps the bacteria to maintain itself in nature. All right, here's another photo of this great gerbil. They're quite charismatic creatures. They appear in the daytime, so we've got to see lots of them. And they're a little bit like meerkats in the way they sort of perch on their legs and watch out for predators. They have different signals for different predators. So birds of prey, there's chirping. If there's a snake around, they'll tap their foot. And they're really quite fun to see. Here's the last picture of great gerbil. I guess I'm trying to persuade you that they're very cute animals, right? All right. So what is this? This is these gerbils, these great gerbils. They live in family groups that consist of maybe a dozen animals, and they're like desert engineers. They build these complex burrow systems in the desert. So this entire thing is one burrow system, and there's maybe 10 or 15 gerbils living in this burrow system. Now, these burrow systems are incredibly important for the gerbil. They will defend these burrow systems to the death. And the reason is these burrow systems, they provide protection from the heat in summer. They provide protection from the cold and the snow and the ice in winter. So this is a desert of extremes, right? You got really, really hot temperatures in summer and really cold temperatures in winter. And these burrow systems, they are central to the gerbil's survival. So they're very, very territorial, and they don't move very far away from their burrow system, partly because of predators, mostly because of predators, and because they want to defend their burrow system from competing neighbours. Now, these burrow systems turn out to be important later, but we'll get to that soon. All right. So, on the vertical axis here, you have numbers of plague cases in humans. This is human plague. This is data on human plague that we're looking at. And on the horizontal axis, you've just got time, and it's just the last century. So what you can see is that in the early half of the 20th century, there were whole villages being wiped out by the plague. It was hundreds of people would die from the plague, and it was quite not every year, of course, but it was a huge public health problem. Now, after World War II, Kazakhstan was part of Russia, and they started a surveillance program. And they're very proud of this surveillance program, and they proudly show this data because it really looks like it worked, right? Surveillance started around 1950, and since then, the number of cases in humans has been really low. The other thing that happened about the time of World War II was the discovery of antibiotics. So when people did get the plague, they started to survive better. So the inset here shows you that drop in the mortality rates around the time of the end of World War II. So plague is easy to treat in humans. You just need antibiotics. So it's no longer the kind of evil threat that it used to be. we talk about Black Death. It had huge repercussions across the world, and there were several pandemics involving the Black Death. We're still sort of scared of it now, right? Black Death is such an ominous phrase to use for an infectious disease, but it actually is a horrible way to die. All right, what am I doing? Yes, so something happened around 1996, and that was Kazakhstan gained independence. It gained independence, and the problem was that the money that used to fund this surveillance program was no longer there, or there wasn't as much money available for the surveillance program. So that's how I got involved. The idea was that with a collaboration between Western scientists and scientists in Kazakhstan, we could find a way to improve the surveillance program so that the country could afford to keep doing it, and they could continue to take public health measures. All right, so let's have a look at what this surveillance actually consisted of. And what they did, it was actually a huge enterprise. They would send out teams of ecologists to different parts of the desert, and they would trap great gerbils. So this is just a simple wooden box trap, and they would put this down one of the burrow entrances. They would go away and wait for a few hours, and when they came back, a lot of these curious great gerbils would have tried to exit the burrow system, and they'd get trapped. So, what's the first thing they do when they catch these animals? They stuff it down a kind of sock. Why the hell would you do that, right? The reason is that there is something about the pressure of the sock on the fur that means the fleas on these animals are triggered to get off. So what you can see emerging from the little gaps in the sock are fleas. Why are fleas important? Well, they're the vector for plague. It's fleas that transmit the plague bacteria from one great gerbil to another great gerbil. And sometimes these fleas transmit the plague bacteria from gerbils to camels or horses or other livestock. And that's the pathway to spill over from the gerbils into the human population. Because what happens to the camel or the cow that gets infected? It dies, and then the farmer will immediately butcher that animal for meat and whatever. But in the process of butchering the animal, they get infected with plague themselves. Anyway, so fleas are the vector, and we are very interested in collecting them and understanding how the movement of fleas might spread plague amongst these animals. All right. Unfortunately, they also kill the gerbils, and they take some of the organs and some of the blood from the animal. And what they do with the fleas and the blood and the organs is they mash it all up and try to grow the bacteria on these agar jelly flakes. So this is what the surveillance consists of. Teams of ecologists catching animals, collecting samples, collecting fleas. It all gets taken back to the laboratory, and in the laboratory they try and grow the plague bacteria. If they can, then they know that plague is active in that area at that time. All right. So what's going on here? When they do detect plague in regions that are close to human villages, human towns, they send out these trucks. And on the back of the truck they've got a pump. And what they do is they stick these rubber hoses down the entrances to the burrow systems, and then they pump insecticide through the burrow system. And that kills the fleas. It doesn't kill the gerbils are fine, but it does kill the fleas. And that's how you control plague. Throughout history, there are examples where people try to control plague by killing the rats during one of these pandemics, people would think it was rats were the culprit, but actually it's the fleas that transmit the plague bacteria. Killing the animals is the last thing you want to do. As soon as you do that, the fleas jump off and look for something else to feed on, to bite. So controlling the fleas is the right way to go. This stops active plague and it removes the public health problem. All right. So this surveillance program, it's been running since the end of World War II, and Russian scientists are very conformist, if you like. They're not particularly creative, and they just did what they've been doing for 40, 50 years. They didn't change what they were doing. If you compare that to sort of the Western style of science, we're always changing what we're doing. So this kind of long-term data set is actually very rare. But let's have a look at what's going on here. We've got the time scale, the horizontal axis going from 1955 to 1995. The vertical bars are prevalence of infection of the Yersinia pestis bacteria. So when you see these vertical bars, plague is active. The solid line connecting some of the dots, that's tracking the abundance of the great gerbils. And what you can see is, there are some gaps in the data, but this vertical axis is the proportion of burrows occupied. So it's a measure of how many gerbils are out in the desert. So what you can see is that there are periods where great gerbils are everywhere. All of the burrow systems are occupied, or most of the burrow systems are occupied. And then there are periods where it's very low. And in fact, it comes down to extremely low values in the early 80s, right? So what you can sort of see is there is a cycling going on. These gerbils, there's a cycle. And actually in the Northern Hemisphere, this is really, really, really common. You have small mammal populations, and they will go through these cycles. Periods of very high abundance and periods of very low abundance. It's not common at all in the Southern Hemisphere. It's not common in Australia. But in the Northern Hemisphere, this is typical. And what you start to see here is that a peak in gerbil abundance is followed by a period of active plague. So this last one is quite a good example. So, the abundance of this great gerbil starts to shoot up, and a couple of years later, you start to get active plague. I'll show you another time series. And this one's even more clear. So it's a little bit rough in the early, in the 50s. The pattern's a little bit rough. But in 1970 you've got high abundance of gerbils, you've got active plague. High abundance of gerbils, active plague. High abundance of gerbils, active plague. So what we tried to do is capture that pattern, and this is where mathematics starts to come in. So far it's all been biology and ecology, but we're going to start to get more mathematical. All right, so the diagram up the top tries to capture this relationship between peaceful gerbil abundance and active plague. And what we see is that if we use as a predictor not this year's abundance, but previous year's abundance, we can start to get a nice relationship between active plague and the great gerbils, what's happening in the great gerbil population. But it's not perfect, right? So when you get to around, what is that, 0.48, in this index of, this horizontal axis is just an index of abundance. It just sort of looks at the previous 2 years of abundance and calculates some sort of weighted average there. But this turned out to be quite a useful predictor of when plague would be active. But it's not 100%, right? It's an abrupt sort of change, but you still get periods, so this is a good example where, you know, the gerbils were everywhere, they were doing really, really well, but we didn't get any play, and they really looked. So this idea that a peak in gerbil abundance is followed by active play, you know, it's not so, there's some noise, right? And that's very typical of nature, very typical that things are very complex and noisy, and we have to deal with that noise all the time. All right, so we've got all this data, and we're starting to form hypotheses about what's going on. And so I thought I'd come back to this diagram to emphasize that we're really in the middle of this green circle. We're at this interface between gathering data and interpreting data. What we want is we want to find patterns in the data that give us insight into how things work. All right, so the question we asked is what causes this abrupt threshold, this change in the system? What's going on? And there's all this theory about infectious disease spreading faster and better when you get high densities. You guys will remember COVID, right? Do you remember that there was a distinction between Melbourne and the rest of Victoria? There was also a distinction between people living in the housing flats in Melbourne and the rest of the city. And that was all based on density of people. The higher the density of people, the more likely you would get spread of COVID. And so the rules and the public health measures were different for the high density populations versus the low density populations. So that's what we thought this was, right? We thought this threshold was, you know, the density of the great gerbils getting higher, so the contacts between the gerbils are getting more frequent, and maybe that helps the fleas get around better. And that's what happened. It's all about contacts. But we started to think more deeply about this, and one thing we noticed was that these burrow systems that the gerbils build, you know, they're quite big, they're quite large. So this is about 20 or 30 meters across, the diameter I'm talking about, 20, 30 meters. And they're quite complex structures. So what you're seeing here is a diagram of one burrow system, and you get a sense for how complex it is. This is, you know, this is depth, so looking at a cross section of the burrow system, and the top one is a bird's eye view of the burrow system. So it's quite amusing how they managed to draw these diagrams and capture this. What they do is they get cement, wet cement, and they pour it into the burrow system. And they wait until they can't pour anymore, and when the concrete dries, they dig up the sand all around the concrete, and that enables them to reveal the structure of these burrow systems. But the consistent thing we started to notice was this bare soil on top of the burrow system. So the plants don't grow very well there, and you just get bare soil. And I remember, you know, this feature, this bare soil effect is pretty consistent across the different landscape types. And I remember one afternoon I was sitting with a colleague, and you know, Google Maps had just sort of come out. You know, I'm old, right? So Google Maps was a novelty, and we were looking at each other's backyards, and one of us, I can't remember who, said, why don't we look at Kazakhstan and see if we can see these burrow systems? So off we go on Google Maps, and this is what we saw. So what's happening is the bare soil is reflecting the sunlight, and the surrounding vegetation is absorbing the sunlight. So you get each of these bright spots here, each of these bright circular areas are burrow systems of the gerbil. And we were absolutely gobsmacked. These animals had colonized vast areas of the desert. Vast areas of the desert. Wherever we looked, we thought we could find them. So what we realized was actually this is a very spatial system. A two-dimensional spatial system. And these gerbils weren't randomly mixing. They're very territorial, so they only really interact with their neighbours. They don't interact with gerbils hundreds of metres away. They're not like human populations at all. So, we're mixing around. I'm from Geelong. Some of you will be from the country. We're mixing all the time. But these territorial gerbils are constrained to stay near their burrow system. That's what protects them from the weather, and it protects them from predators. So we thought, all right, well, maybe what's happening is the plague bacteria sort of spreading in a two-dimensional way through these populations. And there's actually a theory called percolation. So this is just a coffee pot. Sorry. But that's percolation, right? The water has to percolate through the coffee grinds to make the coffee. And if the coffee is too packed tightly, then the coffee will burn. And if it's too loose, then too much water will come through and you get a crap cup of coffee. Right? So percolation is something that emerged in the 50s, maybe earlier than that. And it arose when a statistician asked this question. You know those porous stones, those volcanic stones that have lots and lots of little holes in them? So imagine putting one of those rocks into a bucket of water. Then the question is, can the water get to the centre of the stone? So that's what we call a percolation problem, and it led to a whole branch of mathematics studying this emergent connectivity of graphs, actually. So a simple example of percolation is called bond percolation. And bond percolation or lattice looks something like this. And it almost looks like a maze, so you can imagine trying to get from one side of the lattice to another side of the lattice. But what happens is the more bonds there are, what emerges is what we call long-range connectivity. Actually, it's a really hard topic, so I'm not going to spend too much time trying to explain it. Okay. So we had this idea that maybe this is what's happening, that these burrow systems are like a two-dimensional lattice. The landscape is like this two-dimensional lattice, and the plague has to get from one burrow system to the neighbours, and maybe when you get enough occupancy, if there are enough gerbils occupying the burrow systems. The burrow systems are permanent features of the desert, almost semi-permanent features of the desert, right? And so, occupancy is a really good measure of how many gerbils there are. But you can also imagine that occupancy determines whether something like a plague bacteria can percolate through the landscape, jumping from one burrow system to a neighbouring burrow system, to another neighbouring burrow system, and so on. So, we decide, all right, what we have to do is we have to really check that these bright spots on the satellite image are actually burrow systems. So, that's what we did. We started to gather a new type of data. And so you start to see how mathematics is triggering new questions, new data, new problems. And one problem was we always got stuck. This is our field vehicle breaking down in the desert, and the guys trying to fix it. But we had a great time. We positioned ourselves on on the top of these virus systems and took GPS coordinates, and then matched those up with what we were seeing on the satellite images to make really sure that our interpretation of the satellite image was correct. So the next thing we did was we put together a mathematical model. And what this mathematical model did was brought together three scales. One was the scale of the flea movements between virus systems. So we had marker capture data from fleas, we had marker capture data from gerbils, and so we could see in this data that how far the fleas would typically travel. And it was really, really short. It's only like 100 meters or so at max, at best. So if plague was getting around, it was doing so by making small jumps from one virus system to a close by virus system. The other scale was the scale of the virus system density. So we could measure that from the satellite images. And then the third scale is the scale of surveillance, because this is a vast area of desert, and so our data has a scale associated with it. Right, so we've got this index of abundance here. We've seen that before. And we've got the data. The data we've seen before is these black dots. And these, I want you to look at that black curve. That black curve is the behavior we predicted using this mathematical model. And what we saw was a really good match between the field data, the surveillance data, and the theoretical model. And I was lucky enough to publish this in Nature, which is one of the best journals out there. And I had a great time as well. All right. So one more thing before we stop here. The next question we asked was, well, can we analyze these satellite images and work out which virus systems are empty and which are occupied? Because when the virus system gets abandoned, when the gerbils start to decline, you get regrowth. You get some regrowth on top of the virus system. So we thought maybe we can use the satellite images to measure the abundance of gerbils. And we were reasonably successful. There's quite a few papers on this. And this is a really, what I call a mathematical intensive task to use all sorts of machine learning algorithms to try and classify groups of pixels as virus systems or not virus systems. All right, so what we did was we put these things together. So we've got these satellite images that we can measure the predictor, and we've got this model that uses that predictor to say something about active plague. And this did lead to, it led to more money, which is always good for research, but also improved plague surveillance in Kazakhstan. So that's all I have for today. Thank you.